All right, let's hear it for Jersey City.
Good afternoon. My name is Calvin Weaver, from Jersey City, New Jersey. Here with me is Hafio Kokar, Harshal Agarwal, and Srija Pacha. Our experiment analyzed the effects of microgravity on muscle tissue regeneration compared to an Earth sample. Using an adolescent pig shoulder muscle, we used a medication to help the healing process called tissue regeneration factor, or TRF. We did this experiment because muscle injuries, especially shoulder ones, have become more and more common. In the last year alone, 4.5 million Americans have gone to their doctor just for extreme shoulder pain, including some of our other collaborators who experienced shoulder pain while playing tennis. Hopefully, this data will allow us to understand under which conditions, perhaps microgravity, normal gravity, or even hypergravity environments, under which the muscle tissue will heal the quickest and most effectively.
Okay, for background, we use an adolescent pig shoulder muscle because the anatomy of the pig is similar to that of humans. The tissue regeneration factor includes the main ingredient known as suplexinol, and suplexinol is a bone morphogenetic protein, and these proteins communicate with tissues to promote growth and essentially regenerate injured muscles at a quicker rate. We focused our experiment on the rotator cuff injury because those are the most severe types of injuries, especially in athletes. And we created an incision in the shoulder muscle to imitate that of the rotator cuff injury. And we predicted that the adolescent pig shoulder muscle would work better in space because it has less strain of gravity when it's in space. For our FME lab, the first volume has the TRF-150, the tissue regeneration factor. The second volume is the adolescent pig shoulder muscle, which is within the sustaining solution. The sustaining solution basically provides nutrients for the muscle so it can stay alive. And inside the sustaining solution is the whey protein, the glucose, and the water. And the last volume is the formaldehyde, which is the preservative. When it first reaches the ISS, the first clamp is unclamped so that the adolescent pig shoulder muscle is exposed to the TRF-150. And 2 weeks prior leaving the ISS, the formaldehyde is exposed by unclamping B so that it can act as a preservative.
So to test our hypothesis, we chose to compare the control, which was the FME tube that stayed on Earth, to the experimental, which was the FME tube that went up to the ISS. So now to quantify muscle tissue regeneration, we chose to test for 3 levels, and those 3 levels were glucose, dissolved oxygen, and protein. So all these 3 components are essential for muscle tissue regeneration in almost any living organism. So what our thought process is that if there's a decrease in protein, glucose, and dissolved oxygen, that probably means that theoretically the muscle tissue was able to absorb it and heal to regenerate and fix itself. So aside from doing those 3 levels, we also chose to test for pH because pH is very important to daily medical procedures, and while it's not important to the objective of our experiment, it's important to medical studies and the future of humans in outer space. So that was a quantitative comparison. Now for the qualitative comparison, we also chose to compare SEM images, or scanning electron microscope images, of the test sample to the controlled sample. To test for glucose, we used a calibration curve. So all that a calibration curve basically does is it allows us to translate absorbance readings, which could easily be measured using a spectrophotometer, to ppm or milligrams per liter readings of glucose. So all that means is that we have 5 known solutions with known concentrations, and we know the absorbance of those 5 solutions. So that allows us to create a equation, or a line of best fit, and using that line of best fit, we could translate between the 2 values. Next slide. So similarly for glucose, we had the same procedure for the protein analysis. So also it's a calibration curve with a line of best fit. As for the pH and dissolved oxygen, what we chose to do was we used vernier probes. So vernier probes are simply just really highly selective electrodes. And so we calibrated these electrodes meticulously, and we used standard procedures. So these results for the pH and dissolved oxygen are highly reliable. As for the SEM images, these are the images at a zoom-in level of 30 micrometers. And similarly, these are the same samples, but at a different zoom level of 500 micrometers. So we were able to get these images because of a contact at Montclair State University. So with these images, they're highly inconclusive because muscle tissues are not something that you commonly look at under SEM or a microscope, thus there isn't much precedent about how we go about analyzing these images. So definitely in the future, we plan to reach out to experts in the field to see if there's a way that we could interpret these and make sense of them.
This chart reflects the data that we collected from both FME labs. After the analysis procedures, we found out that there was a higher concentration of glucose and protein inside the Earth tube as opposed to the ISS tube. Now what this shows is that there was more glucose and protein consumed in the ISS tube as opposed to the Earth tube. And in order for muscle tissue to regenerate effectively, it needs to consume more of this glucose and protein. While comparing the dissolved oxygen, or DO, or the pH levels, we diluted the solutions fourfold in order to make our analysis procedures more convenient. And our results, while still inconclusive, support our hypothesis that microgravity enhances the TRF's effectiveness in regenerating this pig muscle. And that's shown by the positive difference between the glucose and protein levels. Since Earth had a greater concentration of glucose and protein, it shows that the ISS tube consumed more of this glucose and protein, as stated before, and therefore it is more efficient in regenerating this muscle tissue. And further studies, we hope, will be conducted in order to reach a more supported conclusion, because there was only one trial for this lab, thanks to SSEP, but if there would be more trials, we could reach a more stable conclusion. And we'd like to extend a special thanks to our teachers, Mr. Stanton and Mrs. Soria, for staying late with us at school in order to conduct our research. Thank you.
Thank you.
Do we have any questions for this team?
Hi, I'm Shanaila, and I'm from Buffalo, New York. I believe that meat has to be alive to regenerate cells, so how did you keep it alive?
So our thought process for doing this is that inside the FME tube, we had all essential components that the muscle tissue would need to be alive and sustain, or be alive for the duration of the trip and making its way back to Earth.
In the central part of the tube with the meat, there was glucose, whey protein, and water to sustain it and allow it to continue to live. So once it is exposed to the TRF 150, it starts healing and continues to do so until the fixative is released into it.
Thank you.